We will wait for another 5 minutes. We will wait for another 5 minutes and then continue. So welcome to the second session of Plants Bioprocessing NPTEL Live session. So in this session, we will be solving some live questions and any doubts regarding your this week 2 session you can ask me here. So that this session mostly will be an interactive session, so I request you all to answer the questions which is being asked, and don't hesitate to ask any doubts which you have. So these are objective types questions only, very simple questions. We are going to solve only 10 questions today, and regarding the doubts which you have asked last week, we will discuss about that also. So the question number 1 is, what is generally done to break the seed dormancy to initiate in vitro cultures? The first option is treatment with phenol. The second is treatment with a dilute acid, treatment with microchloride, and the option D is all of the above. Does anyone know the answer for this question? So what is meant by seed dormancy? Can anybody explain? All of you have gone through the week 2 lectures, right? Okay, so somebody tell me what is seed dormancy? You can type in the answers also if you didn't like to. Yeah, the inability of the viable seed to germinate. Yes, the seeds, they will not be able to germinate. That is one way of describing it. So what of these you will be using it so that it breaks the dormancy period or the sleeping period? Which agent or which solvent or which chemical will you be using to break the dormant period of the seeds? So in dormancy means how to make or break the seed dormancy. So something which will you will be adding that is going to solubilize the layer out. Yes. So it will be acid because it is diluted already, so that it is not going to cause any harm to your cells or the embryo which is present inside your seed coat, but it is going to loosen up your seed coat a bit so that it aids in the germination of your seeds. Okay. So the next question is, the cells of explants which may be induced to undergo division to form a loose mass of undifferentiated cells are known as what? Callus. Yes, callus. Good. Komalavalli, good. Mansi, good. So it is called callus. And why do we call it callus? From where that callus, the word is being adapted for culturing it in vitro? It is like a brush up of your week 2 session, so that you will, so that I will also know whether you guys have understood the topics. While we discuss, whatever doubts you have, you can ask me. So from So does callus happen only in vitro or when we try to generate it, or it is happening in nature also? So you guys told the answer as callus is the undifferentiated mass of cells. And then what is redifferentiation or what is dedifferentiation? What do you mean by those things? What is undifferentiation? Sorry, you guys don't know the answer. So I will slowly ask. So what do you mean by dedifferentiation? Redifferentiation is when cells go back to meristematic phase. Okay. Redifferentiation will be RE, which is redifferentiation is them going back into or they gaining their regenerative ability to form whole plants. Right. What is cytodifferentiation then?
Organ formation.
Cytodifferentiation. That is redifferentiation, right? What do you guys? I think it is in the. It comes in lecture 8 or I don't remember exactly. So what will be, there is a definition will be there na? For Fauzia, what you said was for regeneration.
Organogenesis.
Yes, that is organogenesis. Cytodifferentiation is nothing but when cells which are present in the callus, which is your de-differentiated form, or which doesn't have any, we cannot name what cells there are, but mostly they will be parenchymal cells, right? So when these cells de-differentiate and then redifferentiate, this process will be known as cytodifferentiation. Uh, Do you guys remember this word? Cytodifferentiation? Forget about de-differentiation, redifferentiation. Cytodifferentiation? The answer for this question is callus, but I was asking about the other uh, variations here. So I was talking about cytodifferentiation. Whoever, if they didn't understand what cytodifferentiation is or want to repeat it, you can raise your hands. Yeah. So Komalavali, cytodifferentiation is nothing but in your callus cells, when it is you are keeping an explant and it is differentiating into a calli or what you call undifferentiated mass of cells, which is known as callus, right? So these cells will be mostly parenchymatous. There are many cells like parenchyma, collenchyma, right? So the cells which are from the callus will be more of parenchymatous cells. And these parenchymatous cells, when they de-differentiate and then redifferentiate, this process will be known as cytodifferentiation. It's linked to the parenchymatous cells alone. You're able to get it? Yes, Komagali. So whenever I explain something, if you have any doubts, please ask in the chat box or stop me in the middle also. So this is purely an interactive session where you guys can ask me your questions and doubts, whatever you have related to this week 2 session, and also the previous week's session also you can ask. So last time many of you were asking about bioreactor and these terms were agitation, what is agitation, aeration, those things and all. If you still want me to go into that, please let me know here. So this question is about agitation. So I hope you guys know what is suspension cultures. If not, let me know in the comment box. So the answer for this is D. Yes, Komalvali, the answer is D. So what is agitation and how the agitation is being done with suspension cell culture? Do you know anything about that? How do you give agitation to your cells? The answer is D, which is the aeration. The agitation is helping in aerating the cells, dispersing the cells, making sure there is a better availability of nutrients. The answer is right. The answer is D for this particular question. What I intend to ask is what is agitation, which is basic, and
We are giving some mechanical stress to the cells.
um, mechanical is correct, but it is not stress, Fauzia.
Yes,
Um,
shaking.
yes, shaking. So if you shake something like this, so what do you call it? There will be a rotation of stuff going on there. That will help. That we can in our terms we call it as agitation, or we call it as mixing also. So agitation in case of cell suspension cultures, that will be done by the use of Divya. The answer is D actually. So agitation for cell suspension cultures. Cell suspension cultures means what? The cultures which are inoculated in liquid medium which is kept in flask, your conical flask. It be it 50 ml, 100 ml, whatever may it be, and it will be kept in flask. A liquid medium will be there, and your plant callus cell lines or your hairy roots or whatever your plant cell systems will be introduced into that. So now this flask will be mixed using a mechanical shaker, will be kept inside a shaker. So what will happen? These answers which are listed here will happen. So when agitation is happening by mechanically providing mechanical force inside, this shaker will cause rotations per minute, which we call as RPM, right? Have you come across RPM? It is the unit for rotations per minute it is, or revolution per minute. So this RPM needs to be adjusted so that your cells will get enough oxygen. It will cause aeration to the cells and it will cause a mixing of the cells so that they don't form or stick to the wall of your what you call flask, so that if you keep on mixing it won't, nothing will go and stick to the walls, and also to make sure that there is enough available nutrients to each of the cells present in that particular suspension culture. Okay, so if you have any doubts about this agitation and cell suspension culture, you can raise the hands. If you want me to repeat also, you can raise your hands. No? Okay. Okay. So you guys are aware of auxins and cytokinins, right? So out of this um, options, please choose the correct. Choose what are the auxins. What are the auxins present in this particular question? A and D. Komalavali has said A and D. Shweta is telling A, B, and D. Anybody else? Any other answers? A is alpha naphthalene acetic acid. B is 2,4-dichlorophenoxyacetic acid. D is IA. Yes, the answer is Shweta's, which is A, B, and D, which is alpha naphthalene acetic acid, which is NAA. We call it as NAA. And 2,4-D is 2,4-dichlorophenoxyacetic acid. Indole 3 acetic acid is IAA. Okay, so what is the role of auxins here? Like, what is the role of auxins in plant tissue culture? And first you answer auxin, and then we will talk about cytokinins. They are plant growth regulators, correct? So they are natural, or we are producing ourselves? We are synthetically manufacturing them, or they are natural?
Both
Synthetic.
of them will be. Some will be natural.
Okay, some may be natural. What do you think is the natural auxin which is present in the nature? Out of these, can you name anything?
Auxins.
Yes, natural auxin which is being produced by the plants naturally.
That is,
So this,
I think.
yes, IA is the natural auxin. Correct, correct, Fauzia, correct, Shweta. Thank you for answering. So indole 3 acetic acid is the majorly found auxin, and it is naturally produced by plants. And IBA, which is called indole 3 butyric acid, is also commonly found, but not as major as indole 3 acetic acid, I will say. So the natural auxin among this list is indole 3 acetic acid. And now, what about cytokinins? What do they do? And in this list, can you name any cytokinins?
Keratin.
Kinetin,
BAP.
BAP.
BAP,
Thiadiazerone.
thiodiazerone.
Then
Zeatin.
So all of them are
mhm.
auxins or cytokinins, right?
Cytokinins, BAP, thiodiazerone, etc. I think it is cytokinin.
It is, some people claim it is auxin and some people claim it is cytokinin. This TDC, okay, we will come back to that. So what does auxins do generally? Yes, Komalavali.
Promote shoot growth.
Promote shoot auxin,
Auxin root.
auxin root, and your cytokinin
Shoot.
shoot. So when you want callus, how it will be?
In the
Both
same
in equal ratio.
equal ratio. But do you think that every time that equal ratio works? Say for example, you want a callus of what do you call rose, for example. So if some paper has reported that you have to generate callus, so you need to add just kinetin, any kinetin you can select. Say for example, we will select here B, and we will use auxin as Na, which is naphthalene acetic acid. So if you are doing any experiment, if you add equal concentration, 1 mg per ml of both, do you think callus will come? Might be coming, but not always the case. So if that is the case, what you guys will be doing? What you guys will be doing if you are trying to add, you are adding equal concentration of both any auxin and cytokinin to your media, and you are inoculating your explant. But what you observe is like there is no initiation of callus, not so anything is observed. So at that point, what you guys will do? Any idea? No idea. So the thing is, the equal concentration of both auxin and cytokinin doesn't hold all the time. So if the case which I have explained, like if you even though you go with both equal concentration, if there is no Calais observed, then you have to play around with your concentrations of auxin and cytokinin. Either you can increase it and see, either if you increase auxin and keep the cytokinin same and see whether callus is observed, or decrease or increase the cytokinin and keep the auxin as such, and then do the experiments and check whether the callus is being generated or not. Okay, the scope of this will be explained in your further lectures, where you will be taught about response surface methodology and the process optimization, media optimization and all. Okay, so we will go to the next slide. So the potential of a plant cell to regenerate into an entire plant is termed as what? Totipotency. Yes, totipotency. And what is pluripotent or pluripotency? Ability of a part of the plant to generate into a whole new plant. Mansi has answered. Is anybody? Does totipotency, what is totipotency? Totipotency is the ability of the cells to regenerate into entire plant. What is pluripotency then? I have got only one answer. But it messages the cell to divide itself to perform a certain function. Does it related to something about stem cells or something? Meristematic cells. So pluripotency is as your friend who said, Mansi. So whatever you guys were trying to tell is correct. So actually what it is like, it is the ability of the particular plant tissue or cell to regenerate into any organ. That is what it is about, organogenesis. So Mansi, whatever she said was it's almost right. What else? So, this, what do we guys are, sorry, I have to admit somebody. So we are in plant tissue culture generally, we are extracting which potential of this plant. We are using pluripotency, we are targeting pluripotency or totipotency.
Totipotency.
Totipotency, Mansi, what is your thought in that? Yeah, not there. Okay. So we will go to next slide then if nobody has any answers.
Ma'am, then what
Yes.
will be the definition for pluripotency? Same as that of.
No, 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 they are not same.
Mm,
Because your totipotency is your ability of your cells to regenerate into an entire plant.
okay.
So your pluripotent will be used to what you call I'm not able to get the right words. So your pluripotency will be the ability of the plants is to divide and generate it to a particular organ.
Okay.
So they will not have the entire ability to regenerate. If I'm not confusing, it is, I think it is the right answer, but still I will look and give you the exact definition of the same by the end of the session. Okay? So it's not so confusing. So which of the following developmental process involve an intermediate callus stage while developing somatic embryos from explants under in vitro conditions? Answer. Which of the following is a developmental process which involves an intermediate callus stage while developing somatic embryos from explants under in vitro conditions? Yes, it is indirect somatic embryogenesis. So, what is somatic embryos first? You guys would have come across, right? Somatic embryos, what are they? I think it is there in your, no, it is there in your week 2 lectures. What is somatic embryogenesis? Answer is embryo generation from somatic cells. Okay, what are somatic cells then? Answer is B. The answer is B, correct.
Synthetic seeds, I think.
Synthetic seeds, yes. What are called as synthetic seeds? Why they are named as synthetic seeds? They are named as somatic embryos. What is their purpose? Synthetic seeds, yes, the answer is synthetic seeds. Somatic embryos are synthetic seeds, but synthetic seeds are the ones which we make with somatic embryos. Like we will be encapsulating them and then using them as synthetic seeds, artificial seeds. Mansi, yes, it is vegetative cells. So what there you did you guys have studied? I think most of you are from science background, so I guess you might know what are the different stages of somatic embryo formation.
Can you please explain, ma'am?
Stages of somatic embryo. So, like which is present in our human cells, how the zygote, blastocyst, everything is there. In plant cells also, there are four different stages of somatic embryos. Okay, I will try to draw them. So the first one is the answer for this is this. The first one is zygotic stage, or you call it as like a zygote, like as we have in humans, a round stage. There will be different shapes actually. Like this heart shape embryo. There is actually torpedo stage, there is zygote stage. Somebody wants to join. Then globular stage, where they will form like globules. Then only your 16 cell stage, like what happened here in your what you call before your germination, which is heart shaped. I think this is the torpedo shape. I will give the reference material for this in your drive. Did you guys get the drive link first? I forgot to ask though that. Did you guys get the drive link?
No, ma'am.
No, no, not it. You should be getting it. I don't know why nobody got a drive link for the presentations. Nobody got. No, okay, Divya said no. Okay, then I'll check with the team and let you know when they will be sharing that. Okay, so in that link, I will give the details of this which we are discussing here with reference. I will be giving you. Is it okay? So, what are the advantages of using somatic embryogenesis? There will, for anything, there will be both disadvantages and advantages, right? So, what are the advantages and disadvantages for using somatic embryos? What are the uses of somatic embryos? One you guys said synthetic seeds. Why we have to go for somatic embryos? We can directly grow plants now. No idea? No, okay. Large number of progeny in limited period of time. Yes, that we can tell. What are the other advantages of doing somatic embryogenesis? What about somaclonal variations? What are called somaclonal variations? There will be some advantages of this over in vitro plant generation, right? And disadvantages over in vitro plant generation. One, Shweta has told to produce large number of progeny plants in limited period of time. Okay, that is one. Nobody knows the answers or nobody wants to talk. You guys, to bring uniformity in plant species, yes. What else? No idea. Okay, so what is somaclonal variation then? Or let's talk
The
about,
variation present in the progenies that we created,
yes,
they
there
were
is
not
so many
clones.
genetic
Yeah.
variations will be there while the cells go under each cell division, right? So that will aid in phenotypic changes also. Both genotypic and phenotypic changes will be there. Correct, no? Okay, then we, as many of you were talking about synthetic seeds. What is synthetic seeds? What? How do you do synthetic seeds? Synthetic seeds. Did you study the or did you guys have them in week two regarding synthetic seeds? No. Okay, then when that portion comes, then I will ask that question. Okay, be prepared then. Okay, your assignment is today too, or go and read
Hello,
about.
ma'am.
Yes, yes, Kajal.
Good evening, ma'am.
Yes, Kajal.
Ma'am, actually, I joined this course very late, so I don't know about the assignment. Can you explain me?
No, 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 there is no assignment. I just uttered that words now. No, no need to worry. I didn't give any assignments yet. I am just telling, starting now, so you will be able to understand. So for you guys,
Okay, ma'am.
yeah.
Thank you.
So it is not an assignment. It is just a brush up on thoughts about what are the advantages of using somatic embryogenesis, or what is the advantage of using somatic embryogenesis, and what is the disadvantage of doing somatic embryogenesis. That's all. Just brush up your thoughts on these two advantages and disadvantages of somatic embryogenesis. That's all.
Okay, ma'am.
Okay, just brush those thoughts, but I will be asking the question next week. Let's see who answers first. Okay. Okay, so here again we are in somaclonal variation. So the question is, somaclonal variation is a disadvantage associated with callus cultivation for large scale in vitro production of phytochemicals. Is it true or false? I think with this question, you guys will be able to give an answer on the previous question a bit.
Ma'am, I think it's true.
True. This sentence is true, right? So what will happen? That the answer is true, but uh, how do you think that this will affect the large scale production of phytochemicals? How will soma clonal variation will affect the large scale cultivation of or large scale production of in vitro phytochemicals? The answer is true, but how does this happen? How will this happen?
Ma'am, so many changes occur in the soma clonal variation because pH, temperature all affect that.
Okay. No, what is so that's why the last question I asked in the previous slide was what is homoclonal variation? For this, your friends said that there will be more number of variations in the cells when they divide along. So both genotypically and phenotypically there will be difference. So if there is difference in cells, what will happen? If their gene makeup is different, then the production also will be affected, right? Yes, Vaibavi has given genetic instability might be the disadvantages. Yes, genetic instability is one. Good, Vaibavi. So for this question, the answer is true. True, right? Yes, it is true.
Okay, ma'am.
Yes, Kajal. So the next question is, I think you guys know the answer for this. Which among the following is not a plant growth hormone?
D.
D or B? D.
D.
Okay, betulinic acid is not a plant growth hormone. Okay, good. Fauzia, Mansi, Vaibhavi, and Shweta all have answered. So, what are the others used for? They are plant growth hormones. I can understand, but auxins and cytokinins we already know about them. But all these three, what are they used for? Are they being used in plant tissue culture and plant cell bioprocessing? It's being used or not? Do you guys know? So, what is ethylene used for? Any idea?
It is for fruit ripening.
Fruit ripening, yes. Fruits, you may notice even in your houses, if you keep 4 5 mangoes, raw mangoes along with one mango, what will happen? They will all become yellow in few days because ethylene is in the form of gas. So it will spread and it will cause fruit ripening. We can utilize that in tissue culture also, but the applications will be different. So gibberellic acid, why gibberellic acid is used? We call it gibberellic acid or gibberellin or GA we call as a short form.
Gibberellic acid is used for cell elongation, promotes cell division.
Yes, generally it is used in plant tissue culture for shoot elongation as it has the ability for cell division and cell elongation. If we try to generate in vitro shoots, we will be adding along with your auxin and cytokinin ratio a bit of GA or gibberellic acid will also be used, which will aid in elongation of your shoot. So that is for shoot or cell elongation. So what is abscisic acid used in general? Used for in general? What does obsession means? Shweta Singh has said for flower obsession. What do you mean by that, Shweta? Flower obsession?
I'm inducing more flowering.
I cannot understand. Like you are not able to hear me? Hello, Shweta. So I asked like what do you mean by flower obsession? You have written flower obsession in the comment box, na? So I just asked what is flower obsession. So obsession means what actually?
Related to senescence.
Yeah, delayed. Yes, it is actually the what you call suppression kind of phenomena, growth arrest, or what you call it is opposite of growth promoting kind of stuff. Okay, if you want to arrest something in particular phase, when the people do anther culture, pollen culture, and all, they want to arrest in particular stage or something, they can use abscisic acid. Okay, so the next question is, it's not visible. Okay, I'm going to zoom this out so you can. I, it's not so clear. So okay, so this is a flow chart which shows the development of up to a suspension cell culture. There are some blanks in this. I'm sorry, this picture is not so clear.
Yes, ma'am. The in the second phase, the blank will be sterilization.
Sorry, Fauzi, I cannot. I was not able to hear you. Fauzi, I'm sorry, I'm not able to hear you.
Ma'am, in the first one, the answer will be sterilization.
Okay. What is the second one? Like, give a not the exact word you can. Like, just a, what will be coming. First is sterilization. Yes. So your third point, it is written, place the explant on callus. Okay, the second one is liquid medium. Yes, right. So you are going to cut your callus and put it in a liquid medium or nutrient medium. Yes, absolutely. So what is the third one? Cell suspension culture. From cell suspension culture, there are two. What is third one? This is for, I think this is there in your slides, right? Where your single cell culture methods are being described. Yeah, it's there. So from cell suspension culture, you are going for fine cell suspension culture. So for that, three different techniques are adapted. One is pipetting. The other two are what? Third is filtration. Yes, filtration, correct. What is the fourth one? Addition of what enzyme? What enzyme you will add? So have you heard about the enzyme pectinase? Does it ring a bell? Pectinase.
Yes, ma'am.
Yes, you will be adding pectinase. So, okay, let's start discussing this particular flow chart. So, what is surface sterilization? What are the steps in the surface sterilization? Why do you do surface sterilization? Does anybody have
In order to avoid contamination.
to avoid contamination? Yes, Fauzia. So, does anybody have hands-on training on plant tissue culture here? Have you done any lab or something like that?
We visited the lab but not performed anything.
Not performed anything. Okay. Does anybody else have this access to the lab or able to see what they are doing there? Seen the lab. Mansi also has seen the lab.
Mm,
Okay.
Yes, ma'am.
So,
I have seen, but didn't perform.
okay. So then how do you guys visualize this when I tell surface sterilization?
This washing with running tap water, and after that, 70% ethanol and mercury chloride, etc.
Using
Okay.
laminar airflow chambers or
Okay. So you know theoretically what is happening, but you guys are not aware of how exactly it's being done. Na? I will suggest you
Yes,
guys to
ma'am.
see some YouTube videos. If possible, I will drop in link when in the drive whenever it is being shared with you, so that you can get a picture on what exactly we are talking about when we talk in suspension, surface sterilization, filtration, and all. Okay. So basically, what we will be doing, uh, the first answer is surface sterilization, right? Sterilization. So in your sterilization, you will be bringing your explants. There are two ways. You will be either doing it, if you are doing your in vitro cultivated plants, then is there a need for surface sterilization? It's the plant is already cultivated in vitro in bottles. So do you think you need surface sterilization to be carried out? You guys understand the question? So for any sterilization to happen, there should be a plant material. Then only sterilization it will go. Surface sterilization. My question is, if this plant is already in lab, in vitro produced in bottles, do you think surface sterilization is necessary?
Not all steps, I think.
It's not like that. So why whatever we mean in vitro
even
means
inside lab there might be microbes.
what happened? Komalali, come again. I'm not able to understand your answer.
Even inside the labs there can be any chance for contaminations, right?
You are right. There is chance of contamination, but when you call them in vitro cell system or cell culture, it is bound to be without free from any contamination. That is what we call in vitro plant or something. If there is contamination, we will not be keeping it inside. Okay, only the sterile in vitro developed plants will be used for initiation of callus or anything else. So there is possibility of contamination, like you said, but that is not always the case. When you go in a lab setup, you will understand better. But if you are selecting an in vitro cultured plant which is kept in bottle for your inoculation of callus or induction of callus, you need not surface sterilize it. Only when if your plant is from external source, forest, be it your forest or home or outside your school, college, wherever it is, that plant is staying outside. So it is exposed to microbes from air, water, soil, everything. So these external plants should be surface sterilized. So as you guys said, you will be first washing in running tap water, right? So why do you think we are washing in running tap water? So why do you think we have to wash in running tap water?
If we are taking it from soil and all, there
Yes,
might be.
yes, exactly. So there will be some soil particles, dirt particles will be sticking onto it. Some insects will also be sticking on it, which we may not be able to see properly. Sometimes when you buy your green vegetables and all, you will see small worms and all floating on it, everything, right? So as how we clean your vegetables and fruits at home, same way you are just rinsing it off of any dirt which is present on the surface. Then you can follow all the surface sterilizing protocols which can be established by you, like say for example 70% ethanol or mercury chloride or sodium hypochlorite, whatever be it, so that your end result is to get a sterile explant. Okay. So now you got a sterile explant and you have put it in one solid media. Say for example in a plate you have put. So what will happen? Your media is callus initiating media. So what is your expected outcome from that? You have a sterile explant and you are putting it in a media which will induce callus. What will be the outcome? What will be the outcome? I gave the answer half. Your media is a callus inducing media and you have a sterile explant. The end result is what? What do you expect in 20 days or 30 days from that media and the explant?
Mass of cells.
Yes, we will get a callus, right? So you will be getting a callus. This is what is this step. After you do all these surface sterilization protocols, everything, cutting them and then keeping it in a media of choice, you will be getting a. If you keep it in a callus induction media only, you will get a callus, not in some other media. If you have a shoot replication media and you are keeping the explant on shoot media, shoots will come, not the callus. Understood? So now you got a callus. So callus will be what? It will be like this, na? Somewhat undifferentiated. It will not have a defined shape or structure to it. It will be of whatever kind it is. So now you are going to take portions of this. That's why it is written chop or cut callus and put it in liquid medium. So what before this step comes, this callus will be multiplied multiple times. It will be transferred. What is mean by subculture? Can any you guys will be working on microbial stuff and all now? What is subculture? Why do we do subculture?
Reculturing from the already existing culture.
Yes. What is the purpose for doing that? Why do you do that? You are reculturing it, understandable, but why you need to do? Why do you have to reculture it when it is already existing in a media? Why do you have to reculture it? So you are reculturing it.
To maintain the nutrients.
Yes. Yes, Shweta, it is to provide proper nutrients because if it is exhausting the nutrients which is already present, say for example, you have kept it in one plate and left it for 10 days. Maybe it has utilized all the food which is present in that particular plate. So you need to transfer, cells will not transfer them themselves now. So you have to transfer them to a new media so that they will get further food, new food. Same media composition, but it should be kept on changing. So if you do so, like how we grow as we are, we keep on having proper food and over the years we start growing, right? Same way your cells also will start growing further. If it is 1 gram here, maybe in the next subculture it will increase its weight to 2 gram. Then in the next subculture, it is not 1 gram increase all the time. I'm giving an example. So the cells will also multiply when you subculture things. Understand? So once you get a multiplication, say for example, you need 5 grams of callus or something. You need to establish some 2 to 3 subcultures. Then you will get the mass of cells which you want. From this cells, you can take a bit of it, cut it, and transfer to your liquid medium. So from many subcultures, your cells will be multiplied and then will go into your suspension culture. Okay, so here is your cells. So, okay, first we are
Ma'am,
initiating. Yes,
excuse
yes,
me.
yes, Fauzia.
Ma'am, is there any ways to or procedures to cut it in to cut and inoculate in next medium?
There are no, when you do practically, it depends on the hands actually. So there are no written rules on
Okay.
this is the way you have to cut, not like that.
Okay.
Okay, so say for example, if your callus, you have come across, na,
Mm-hmm.
ma'am would have thought about compact callus and friable callus, right? Have you come across that?
Mm.
Yeah. So compact callus will be stiff. It needs to be cut because otherwise you cannot separate them into pieces. They will be stiff. So you have to use forceps and
Okay.
scalpel to cut them a bit and bit pieces. So this hard portion, maybe circular or whatever shape it will be very hard, compact one. So you have to cut them into some particular, there is no particular shape or size you want to cut, okay? So this will go into the media. In case of friable callus, they will be very, very friable. They will be like slimy, thin, small cells. Okay. For that, you need not cut into anything. You can just scoop up and put. It is like scooping them up using a spoon or spatula. You can just scoop them up and put into another fresh media so that it is transferred to the fresh media. So here, in case of this place the experiment, chop or cut the callus and put it in the culture. If it is hard, compact callus, you are going to chopping it and putting it in the suspension culture. If it is fine callus, you are just scooping it out and putting it in your liquid media. Understood? Able to get it? Yes. So,
Ma'am,
yes, Shweta.
ma'am, could you please explain why we are using pectinase enzyme?
Yes, I will come to that point. Now I'm in this now, I will come there. Okay. So now you guys have
Okay, ma'am.
yes, thank you. So now we guys have initiated cell suspension culture. So you have cut and put your cells, sorry, in your conical flask. So the cells, like you said in the earlier questions, agitation will be given so that cells will be getting equal nutrients and aeration, everything. And what will happen? The cells will start growing, right? So you have cut and put your cells. The cells will have fine cells also and aggregates also, right? Due to your mechanical stress, it will be broken down into small ones. So there will be finer cells also. There will be cells of different sizes because your aggregate size also varies. A small lump also will be there. Bigger lumps also will be there. It varies. Okay, that is the reason you are going for generating a finer cell suspensions. So the answer here, which is third one, what was the answer? You are filtering it. The end result which you want is like, say for example, the from the cell suspension. Let me go to the next page. So this is your cell suspension. Here aggregates and all is there. Okay. So you are allowing it to grow. Now it has grown up to media will become less, cells will be more. This is how the growth will be after how many days. It depends on, it varies according to the plant species. So now you will be needing finer cells. To get finer cells, filtration you can do. In filtration, what you will do? So you can use a funnel with the filter size and filter paper size and all varies. So if in case, say for example, you are using zero point 0.4 micrometer filter, means if it filter this filter membrane, through the membrane, if your cells you're pouring like this. Say let it be like this, okay? So this size, this filter's hole, pore size. Pore size means what? The size of the holes in that filter. So if you have seen filters, it will be filter paper, or just for explanation sake I'm putting here. So each hole size will be around, pore size is around 0.4 micrometer, means the cells which are passing through it also will be what size? More or lesser than or equal to 0.4, na? Are you able to get the point? So
Yes, ma'am.
any larger clumps will be settling on top of your filter membrane, and anything which is smaller than the size of the pore and smaller than or equal to the size of the pore will be passing down through a new flask. Here you will have your new culture media. So this finer cells will be coming here. So this will have your fine cells. So this is called fine suspension, fine cell suspension. This is one way. So what is the other way of doing it? Pipetting. So in pipetting, what will you do? There will be fine cells also. You cannot pipette fine cells out and through. What you can do, you can pipette out the larger lumps, or aggregates. You can pick in your flask, this flask, you can use a pipette to take this large lump and transfer it to something else, so that whatever visible aggregates are present in your media is being removed by pipetting. Okay, that is another way. So coming to the pectinase. So to understand what is the role of pectinase here, we should know what is pectin mean. What is pectin? I guess you guys will know pectin. Where pectin is there in plant cells. Pectin will be
It is
there.
the outermost
Yes, it is in the
covering.
cell wall. It is in the cell wall. Or it is a component of the cell wall. So, if pectinase means that particular enzyme has the ability to disintegrate or degrade pectin, right? So now tell me what function it would have on making the cells as a fine cell suspension. So pectinase is an enzyme. You guys came to that point there. It will degrade pectin. This pectin is found where? On the outer surface or outer cell wall. It is a cell wall component of plant cells. So what is going to happen? Your aggregates will be like this. So if your pectin is going and disintegrating this junction, the cells are attached by what? They are attached to each other like this, na? They are on top, here, there, and all. So this junction will be ruptured if you add pectinase. So what you will get? You will get finer cells, right? More cells are sitting on top of each other, then that is what is called as aggregates. So if you add pectinase, they will all disintegrate. They will become single cells instead of being as a club, or together. Instead of being together, they will become separate. So that is the purpose of using pectinase. Shweta, you are able to get that? Are you able to understand? Yes. So this will be the role of pectinase. There are other enzymes also. You guys, next week when we come for the next lecture, there are two things I want you guys to do and come. One is you have to tell me the advantages and disadvantages of somatic embryogenesis, and the second one is is there any other enzymes being used to make cell aggregates into single cells or disrupt this aggregation. Pectinase is one. Is there any other enzymes? You have to check and come. Okay. So I think yes, I think this is the end of the session. If you have any doubts, you can ask now. Hopefully, the drive link should be reaching you by Monday. We will see. If not, I will drop my email ID here. If not, please mail me. Any doubts? Do you guys have any doubts? So last week you guys asked for any books, right? So I will, once you get the drive link, I will be uploading it in the drive, and some plant tissue culture videos or something so that you can go through, or you can just type in YouTube plant tissue culture, then you will get lots of videos. There is, I think, one channel called Plant. I don't remember exactly. I think that channel name itself is Plant Cell Technology or Plant Tissue Culture, I think. If you type that, you will get a plenty of videos on that particular topic so that you will get an idea on what exactly they are doing. So I think uh, everybody has got their. What about your assignments? Have you all submitted your first assignment? Second assignment is due when?
I think first assignment due date is on 7th or
Okay,
something
you
like.
guys yet to submit it. Okay then. So if you have any doubts, you can ask me here. Second one is on August 9th. Okay, Shweta, thank you for letting me know. So if you have any doubts, you can ask me here, or if you are done with your doubts and you don't have anything to ask, you can leave the session. Thank you for joining.
Ma'am,
Yes, Kajal.
in first assignment, what we have to do?
I have not seen your assignment, so I have no idea.
Because ma'am, I joined this course very late. That's why I don't know.
No, you joined this live session late or the course?
Live session.
Live session pay, there is not a big assignment and all is there. There is no assignment. I have asked two questions which nobody answered. Okay. So I just wanted you guys to go and get the answer for that when you guys are coming for the next live session. So the first question is, what are the disadvantages and advantages of somatic embryogenesis? Second question is there any other enzyme is available to make fine cell suspension? Those
Okay,
questions.
ma'am.
These are the two questions. Don't think about assignment. This is for you guys to brush up on your thoughts about this particular topics because I was not able to, nobody was answering. That's why. Okay.
Okay, ma'am. Thank you.
Yeah, thank you. If you have no doubts, you can leave the call. Thank you for joining. ஆ, ஆமா. எனக்கு கிளாஸ் இருக்குல்ல சார். என்ன பேசிட்டு இருக்கீங்க? முடிஞ்சது. ம். கொஞ்சம் தான் சாப்பிடுவேன். Bye. ம். Bye bye. எங்க இருக்கடா?
Has the class ended?
Yes, Mansi? What you're talking? Like, hello? Mansi, I'm not able to hear you. Can you repeat? Hello? Mansi, am I audible? Joey, the session has actually got over.
Okay, ma'am. Okay, ma'am.
Yeah, if you have any doubts, you can ask.
No, ma'am, I don't have doubts.
Thank you for joining the session. We will meet you next time, next week, the same time, 6 to 8, to discuss about the week 3 lecture sessions. Thank you for joining. Thank you for joining. So we will be meeting next week to discuss the week 3 questions. And if you have any doubts, please reach out to me in the mail ID given. So thank you for joining this session. We will meet you next week.